Nucleic acids are one of the four major classes of biological macromolecules, alongside proteins, lipids, and carbohydrates. Their primary role in living systems is to store, transmit, and express genetic information. In other words, nucleic acids carry the instructions that tell cells how to grow, function, and reproduce. There are two main types of nucleic acids, DNA and RNA, and each plays a distinct but closely connected role in the flow of genetic information within the cell. DNA, or deoxyribonucleic acid, serves as the long-term storage of genetic information. The sequence of DNA encodes the instructions needed to build and maintain an organism. This information is passed from one generation to the next, making DNA the molecular basis of heredity. To preserve this information, DNA undergoes replication, a highly accurate process in which the DNA molecule is copied before cell division. This ensures that each daughter cell, or each offspring, receives essentially the same genetic instructions. RNA, or ribonucleic acid, is primarily involved in the expression of genetic information. RNA molecules are produced from DNA through transcription, and many RNAs participate in translation, the process by which genetic information is used to synthesize proteins. In this way, RNA acts as the functional link between stored genetic information and cellular activity. Because genetic information is essential for survival, cells have evolved mechanisms to minimize changes in DNA sequence. Errors can occur during replication or result from environmental damage, but these changes are limited by specialized DNA repair mechanisms that detect and correct damage, helping maintain genetic stability over time. DNA functions as a molecular information system for the cell. The sequence of nucleotides in DNA encodes instructions that guide cellular structure, activity, and behavior. This information is not used directly to perform cellular work, instead, it is interpreted and executed through RNA and proteins. The information stored in DNA is first transcribed into RNA, and many of these RNA molecules are then translated into proteins. Proteins carry out most cellular functions, including metabolism, signaling, structural support, and regulation. In this way, DNA provides the blueprint, RNA acts as the intermediary, and proteins serve as the functional output of genetic information. Because DNA encodes functional molecules, changes in DNA sequence can lead to changes in cellular function. A modification in a gene may alter the sequence of an RNA or protein, which can affect how well that molecule functions or whether it functions at all. Some changes have little to no effect, while others can significantly impact cellular processes. The relationship between DNA sequence and observable traits is described by the concept that genotype determines phenotype. The genotype refers to the genetic information encoded in DNA, while the phenotype refers to the physical, biochemical, or behavioral traits that result from that information. Differences in DNA sequence can therefore lead to differences in phenotype. During DNA replication, rare errors can occur, resulting in mutations. These mutations may change the amino acid sequence of protein products or alter gene regulation. While some mutations can be harmful or neutral, others introduce genetic variation that contributes to diversity within populations. This genetic variation is a fundamental driver of evolution. Mutations that affect protein function or regulation can influence an organism's fitness, and over time, natural selection can act on these variations, shaping the genetic composition of populations. The primary structure of nucleic acids refers to the linear sequence of nucleotides that make up a DNA or RNA molecule. This sequence is the most fundamental level of nucleic acid structure and is where genetic information is encoded. Each nucleic acid is composed of repeating units called nucleotides. A nucleotide consists of three components: a 5-carbon sugar, a phosphate group, and a nitrogenous base. The sugar is deoxyribose in DNA and ribose in RNA, while the phosphate group links nucleotides together to form a long chain. Nucleotides are connected by phosphodiester bonds, which form between the phosphate group of one nucleotide and the sugar of the next. This creates a sugar-phosphate backbone that gives nucleic acids their structural stability. Because of this linkage, nucleic acids have directionality, with a 5' and a 3', which is essential for replication, transcription, and translation. The sequence of nitrogenous bases along the backbone adenine, guanine, cytosine, and thymine in DNA, or uracil replacing thymine in RNA constitutes the primary structure. It is this specific order of bases that encodes genetic information. Even small changes in the primary structure, such as a single nucleotide substitution, can alter the information content of DNA or RNA. These changes may affect how genes are transcribed or translated and can ultimately influence protein structure and function. Thus, the primary structure of nucleic acids provides the molecular basis for information storage, serving as the foundation upon which higher-order structures and biological function are built. DNA and RNA differ significantly in their chemical stability, and these differences are closely tied to their biological roles. DNA is a highly stable molecule, which is essential for its function as long-term genetic information storage. One key factor contributing to DNA stability is the presence of deoxyribose, which lacks a hydroxyl group at the 2-carbon. This absence makes DNA less susceptible to chemical breakdown, particularly hydrolysis. In addition, DNA is typically double-stranded, with complementary base pairing and hydrogen bonding between strands. This double-stranded structure protects the genetic information and allows damaged regions to be repaired using the complementary strand as a template. DNA stability is further supported by cellular repair mechanisms that continuously monitor and correct damage caused by replication errors, radiation, or chemical exposure. Together, these features allow DNA to preserve genetic information over long periods and across generations. In contrast, RNA is generally less stable than DNA. RNA contains ribose, which has a hydroxyl group at the 2-carbon. This chemical feature makes RNA more prone to hydrolysis, especially under alkaline conditions. As a result, RNA molecules are more easily degraded. RNA is also usually single-stranded, which contributes to its reduced stability. Many RNA molecules are designed to be short-lived, allowing the cell to rapidly adjust gene expression in response to changing conditions. These differences in stability reflect functional specialization. DNA serves as a permanent information archive, while RNA functions as a temporary and dynamic messenger that enables flexible regulation of cellular processes. The secondary structure of DNA refers to the way a DNA molecule folds and organizes itself beyond its linear nucleotide sequence. The most well-known and biologically relevant secondary structure of DNA is the double helix. DNA consists of 2 polynucleotide strands that run in opposite directions, a feature known as antiparallel orientation. One strand runs from the 5' to the 3', while the complementary strand runs from 3 to 5. This antiparallel arrangement is essential for base pairing and DNA replication. The 2 strands are held together by complementary base pairing. Adenine pairs with thymine through 2 hydrogen bonds, while guanine pairs with cytosine through 3 hydrogen bonds. These specific interactions ensure accurate information storage and copying. The sugar-phosphate backbones of the 2 strands form the outside of the helix, while the nitrogenous bases are stacked on the inside. Base stacking interactions, along with hydrogen bonding, contribute significantly to the stability of the double helix. In most cells, DNA adopts the B-form double helix, characterized by a right-handed twist, uniform diameter, and regular spacing between base pairs. This structure creates major and minor grooves along the helix, which serve as binding sites for proteins involved in transcription, replication, and regulation. The double helix structure is not only stable but also functional. It allows genetic information to be compactly stored, accurately replicated, and efficiently accessed by cellular machinery. The complementary nature of the strands ensures that each strand can serve as a template for the synthesis of a new strand during replication. While the DNA double helix is the most familiar secondary structure, nucleic acids can adopt a variety of alternative secondary structures depending on sequence, environment, and function. In DNA, variations of the double helix can occur. In addition to the common B-form DNA, DNA can also adopt A-form and Z-form structures. A-form DNA is shorter and wider and is often observed in DNA-RNA hybrids and double-stranded RNA. Z-form DNA is a left-handed helix that forms in regions with high GC content or under specific ionic conditions and may play roles in gene regulation. RNA molecules display even greater structural diversity because they are typically single-stranded. RNA can fold back on itself through intramolecular base pairing, forming structures such as hairpins, stem loops, bulges, and internal loops. These secondary structures are stabilized by hydrogen bonding between complementary bases within the same molecule. These RNA secondary structures are not merely structural, they are often essential for function. For example, transfer RNA, tRNA, folds into a characteristic cloverleaf secondary structure that is critical for accurate translation. Ribosomal RNA, rRNA, forms extensive secondary structures that contribute to the catalytic and structural roles of the ribosome. Certain nucleic acid sequences can also form specialized secondary structures, such as G-quadruplexes, which arise in guanine-rich regions of DNA or RNA. These structures can influence replication, transcription, and genome stability. Overall, alternative secondary structures expand the functional capabilities of nucleic acids, allowing DNA and RNA to participate in regulation, catalysis, and dynamic cellular responses beyond simple information storage. The structure of the DNA double helix is reversible, meaning the two strands can separate and rejoin under appropriate conditions. This behavior is described by the processes of denaturation and annealing. Denaturation refers to the separation of the two DNA strands due to the disruption of hydrogen bonds between complementary base pairs. Importantly, denaturation does not break the covalent phosphodiester backbone, it only disrupts the non-covalent interactions that hold the strands together. One common cause of denaturation is elevated temperature, a process often referred to as DNA melting. As temperature increases, thermal motion destabilizes hydrogen bonds and base stacking interactions, causing the double helix to unwind and separate into single strands. When denaturing conditions are removed, complementary DNA strands can reassociate through a process known as annealing or renaturation. Annealing depends on how much of the molecule remains base paired. If part of the DNA molecule is still base paired, annealing is typically a rapid, one-step process, as the remaining paired region serves as a guide to quickly reform the double helix. In contrast, if the two strands are fully separated, annealing occurs more slowly. In this case, the strands must first find each other by random collision, which is a slow, rate-limiting step. Once a complementary region aligns and base pairing begins, the remainder of the helix reforms rapidly. These principles of denaturation and annealing are fundamental to many biological and laboratory processes, including DNA replication, transcription, PCR, and hybridization-based techniques. The denaturation of DNA can be quantitatively analyzed using denaturation curves, which describe the relationship between temperature and the extent of strand separation. As DNA is heated, the double helix gradually unwinds, and base pairing is disrupted. This process can be monitored experimentally by measuring UV absorbance at 260 nm. Single-stranded DNA absorbs more ultraviolet light than double-stranded DNA, a phenomenon known as the hyperchromic effect. As a result, absorbance increases as the DNA denatures. A denaturation curve plots UV absorbance versus temperature and typically shows a sharp transition from double-stranded to single-stranded DNA. This reflects the cooperative nature of DNA melting, in which regions of the helix unwind together rather than independently. The melting temperature, or TM, is defined as the temperature at which 50% of the DNA molecules are denatured and 50% remain double-stranded. TM is a useful parameter for comparing the stability of different DNA molecules. Several factors influence the melting temperature of DNA. GNC content has a strong effect on TM. Guanine-cytosine base pairs form 3 hydrogen bonds, compared to 2 in adenine-thymine pairs, and also exhibit stronger base stacking interactions. As a result, DNA with higher GC content generally has a higher TM. Solvent conditions, including the chemical composition of the solution, can alter DNA stability. Organic solvents or denaturants can weaken base pairing and lower the TM. Salt concentration plays an important role because positive ions shield the negative charges on the DNA phosphate backbone. Higher salt concentrations reduce electrostatic repulsion between strands, increasing stability and raising the TM, whereas low salt conditions decrease TM. pH also affects DNA stability. Extreme pH conditions can disrupt hydrogen bonding and alter base structure, leading to strand separation and a lower TM. Finally, the length of the DNA molecule influences melting behavior. Longer DNA molecules generally have a higher and broader melting transition because more base pairs contribute to overall stability.